Hello and welcome everyone to this podcast. And in this podcast, we're going to talk about nuclear organization and how that links to gene expression. And then as we're talking about it, we're going to talk about how epigenetics feeds into this. Up to now, we've talked a lot about epigenetic control of gene expression. And ultimately, the goal of controlling gene expression using these modification mechanisms is to precisely control gene product output. Which genes are going to be turned on at the right time, in the right tissues, at the right time of development. And in doing so, we've talked about a few things here. We have a gene of interest here that we want to determine if we're going to turn it on or off. We've talked about how we can add methyl groups to the DNA, and in doing so, that would turn the gene off. We also talked about how we could modify various histones that are in this region. And we talked about several different things. We've talked about H3, K4, H3, K9, H3, S10, and a few others. And in talking about these, we've talked about how they could be methylated, phosphorylated in the case of S10 up here, or acetylated. And we talked about different mechanisms to bring these groups in, different mechanisms to maintain the epigenetic modification, and how that ultimately leads to a difference in gene expression. However, that's not the whole story. There are some other factors that we want to talk about. So some other factors not linked directly to the modification of the genome here. We also want to consider genes' relative position in the nucleus. We also want to talk about the proximity to the nuclear membrane. And last, within the nucleus, we're going to talk about how we can incorporate regions of the DNA into things we call transcription. I'm going to abbreviate it because I'm running out of space here. Transcription factories. So as we move forward, it's going to be important that we understand the basic architecture of the nucleus. So let's spend a little time talking about the nuclear architecture. We used to think that, if this were our nucleus here, that the biggest problem was just kind of getting all that DNA into the nucleus. And then once it was in there, it would kind of work its way out. So there's kind of this like random packing. It was efficiently packaged because you had to get a lot of DNA in there. But the goal wasn't to pack it into the nucleus in a usable way. What we've learned is that that's not true. How could that possibly be true? But rather, what we now accept is that the structure of the nucleus, inside the nucleus, is highly organized. It's a highly organized structure. But it is also dynamic, meaning it will change states throughout a course of a cell's life. This highly organized structure here is the nuclear architecture. But let's give it a more formal definition here. Your book describes it as such. It's these morphological and functional heterogeneity that is formed by positioning of different subunits or subunit compartments in the nucleus. So what that means is that, as we'll see in a moment, that as chromosomes are placed into the nucleus, they're placed in a very precise manner, but you can see that there's a difference. There's going to be a difference in this region here versus this region versus this region. So that gives us what we might call heterogeneity. These compartments that I'm talking about here, we can place into one of two categories or types. One would be nuclear bodies, and the second would be chromosome territories. So let's consider some of these nuclear bodies and chromosome territories. So let's first talk about nuclear bodies. And we can define a nuclear body by a distinct subnuclear region that lacks a membrane. But even though it lacks a membrane, These nuclear bodies are characterized by specific proteins and DNA regions in some cases. Let's talk about some examples. And this first one that we're going to spend quite a bit of time with is called the nucleolus. To remind you, the nucleolus is a place where ribosomes and ribosomal RNA are made. So let's draw a nucleus over here. And I'm going to draw this first nuclear body in here, the nucleolus. Again, even though I drew a circle around it, there's no membrane around it. It's just the defined region within the nucleus that has a very specific function. The next one I want to mention are these things called Cajal bodies. This is where small ribonucleoproteins are made. We call these SNRNPs. These play an important role in splicing of the messenger RNAs. They're often found near or associated with the nucleolus. Not in it, but near it. And I'm just going to put CB here for Cajal bodies. The next one I want to talk about are PML bodies. These PML bodies largely have an unknown function, but the name PML stands for promyelocytic leukemia proteins. And what they've observed is that these PML proteins will form these very concentrated structures in the nucleus. And lastly, we have thousands of these transcription, TXM for short, transcription factories. These are areas where RNA polymerase II are highly localized, and it's areas within specific regions of the nucleus that are showing high transcription levels. And I'm going to show these in here in just a moment. I just want to identify a couple other things before I show that. Okay, I'm going to keep this picture going here for now, but I need to make some room to talk about chromosomal territories. So let me talk about chromosome territories. Chromosome territories are the organization of each chromosome in the nucleus. How it's organized is very specific for that particular chromosome. And within this chromosome territory, there will be some variability on where different genes are localized based upon their gene activity levels. So for instance, I'm going to draw a few here. They're going to have different shapes to them based upon how big the chromosome is. And you can see I'm wrapping them around these other regions because they are distinct from each other. So these would all be chromosome territories. I said I'd come back to these transcription factories, and I will do that now. Within these different chromosomal territories, there will be regions that are going to be highly transcriptionally active. There might be larger, smaller regions, but this is where you're going to see RNA polymerase IIs. And I write that down here. RNA polymerase II. So I want to spend a little bit more time talking about these chromosome territories. Remember, within these chromosome territories is all the DNA. And there's going to be regions here, and I'll attempt to draw it here as very tightly packaged regions. So we have our DNA, and then we have some regions that are highly compacted within this region here. And this would be our heterochromatin. If you noticed, I intentionally left spaces between these chromosomes. Between these territories. And these are just channels, nuclear channels. And this allows for the transportation of large protein complexes to different territories if necessary. Within these territories, I'll just point out a couple more things here, but there would be definite areas where we could see the centromere, and other areas where we'd have the telomeres, the ends of the chromosomes. So each of these territories is very distinct, contains the entire chromosome. Now, even though they have these very specific regions here, it's not uncommon for some of the DNA to loop out and maybe interact with other loops. It might be that these two regions share a common transcription factor and they can be co-regulated at the same time. So it's not uncommon to see them pop out like so. Okay, the next thing I want to talk about is the nucleoskeleton. The nucleoskeleton contains many fibrous proteins that participate in DNA activity and packaging, and can serve as a scaffold to hold that DNA in the right proper places. Okay, I'm going to erase this picture over here because I need to draw some features of the nucleus skeleton, and I think it'll get too messy if I keep on this one figure. So let me draw our nucleus again. And I haven't really been drawing it this way, but I'm going to just so we remember that the nucleus itself has two membranes. The proteins that I mentioned in describing the nucleus skeleton are by and large proteins we call lamins. These lamins are often found associated with the inner side of the nuclear membrane. And what they will do is they'll serve as attachment sites. If this is a large chromosome in this territory here, it might loop up here, it might loop up here or so, and it'll attach to these lamina proteins. There can also be some internal laminins that will bind to the DNA on the inside of the nucleus. It's also not common, as that is looping out there, for it to interact with our nucleolus. So that's our nucleolus. And you can see this loop coming in here. We're going to come back to that in a moment. But a little bit of a preview. We're going to see that almost all the chromosomes have a loop that extends into the nucleolus. But it's directed there by these laminin proteins. And I'm going to kind of extend some of this chromosome out some. And what we'll see is that within these regions here, we'll have heterochromatin. And we will also have regions, say right here maybe, which we will call transcription neighborhoods. Clustered at this site here are the various transcription factors, epigenetic modifying proteins, all in this area here to help stimulate transcription in this very specific region. Chromatin here, being very flexible, as we'll see in a moment, can allow for the association of pieces of DNA that are actually distant-wise quite far apart, but now they are close to each other. Okay, there are two types of lamins. The first type we'll call type A, or rather A type. The A type are generated from the gene LMNA. That's the gene. And the protein that's made from this would be lamina A or C. The next type is B type. The gene that makes the B type laminins is called LMNB1, and there's another one called LMNB2. And they make the proteins laminin B1 and laminin B2. Let me say a couple things about a few of these here. Laminin B1, so laminin B1, it's important in nucleoskeleton integrity, and it's important in gene expression. We know this because if we mutate laminin B1, we no longer form these loops, no longer these attachment sites to the outside of the nucleus here, or the most inner side of the nucleus, I should say. And we see reduced gene expression. So the mutant, we don't see loops, so no loops, and reduced gene expression. Now I want to say something about laminin AC. We believe laminin AC plays a larger role in scaffolding. We know that there are several disorders that are linked to mutations of laminin AC. So disorders linked laminin AC mutants. The next thing I want to talk about are these things called active chromatin hubs. And what I'm going to show you, this is sort of the loop we just had, but I'm going to kind of exaggerate it here a little bit. Remember, the DNA is very flexible. As I had on the previous slide, we see areas of heterochromatin. So, of course, reduced gene expression here. We may have other genes here that are not in heterochromatin, but still not expressed. I'll explain why in a moment. We then may have a gene here that is expressed. And the reason it is expressed is because various transcription factors and chromatin modifying enzymes associate with these two regions of the chromatin. Now, distant-wise, they're a fair bit apart, but they are close to each other now. Okay, so these are regulatory proteins that have been brought close to each other. And that allows this gene to be expressed. Now this gene can't. Now maybe under a different circumstances, the chromosome would flex in a slightly different direction, and now this gene would be closer to some activator sites and activator proteins. Now one thing I want to say here is this is one way that a family of genes that might be close to each other, and one example of this would be the globin genes. We may or may not talk a little bit more about these. Globin genes or the Hox genes. This may be a way to regulate which globin gene or which Hox gene is turned on if you can put one in front or in adjacent vicinity of these regulatory proteins. Whereas this one here, if it's a member of the same gene family, it's not turned on. Okay, I'm going to talk one more thing about these loops, but I'm going to talk about it in its relationship to forming the nucleolus. So let's talk about forming this nucleolus. And if you remember, if this was our nucleus, the nucleolus formed somewhere in here. I also showed you on an earlier board these chromosome territories. And I talked to you about just recently then how the DNA in here can form these loops. And sometimes they can extend from the territories. And that's what we're going to draw here. Within each of these chromosomes, there are several genes that we'll call rDNA. There are genes on the DNA that code for R or ribosomal RNAs. By having those genes, which are not adjacent to each other, they're in different parts of the chromosome, loop out here. So maybe there's one here. I'll just put a 1, a 2, a 3, and maybe there's a 4th one here, 5th one, 6th one, 7th one, in these various locations. And actually, the way I drew it is pretty simple here, but it can be much more complex. If you had a ribosomal gene down here, it would have to find a way to loop out into this open space here. This is a way to get all of the ribosomal genes in the nucleolus, or in this structure that will become the nucleolus. Activating or transcribing these genes requires a different RNA polymerase and other factors that are different than turning on genes in other areas. So it's important to get them all in one area. We can think of the nucleolus. If I ever slip and call it the nucleosome, forgive me, it's supposed to be the nucleolus. So this nucleolus forms by the establishment of chromatin loops containing rDNA. Now, how does it know to do that? Well, that's a good question. I'm glad you asked. What happens is there are these regions here that I put in this purple square here that we call nucleolar organizers. And really what they are is just the DNA sequences that form tandem repeats. Just specific DNA sequences here. And what we see is we see these tandem repeats here and there, all right at that border. Now within the nucleolus, I'm just going to draw kind of this, I'm going to try to draw it like a circle here where it's touching each of these loops here, and it would be touching these other loops here. I just left a lot of space here just so it wouldn't get too complicated in here. But this here that I drew here in pink is something we call the fibrillar center. And the fibrillar center holds these ribosomal genes, the rDNA, in place within this newly formed structure, the nucleolus. So the question I want to address now is, how is this linked to epigenetics? Well, one way is that we are localizing genes here for activity. So we are localizing genes for common activity. These are ribosomal genes. So to transcribe them, we will need transcription factor 1, and we'll need RNA polymerase 1. So by bringing them all in this one place, we can concentrate the proteins and the enzymes necessary for those genes. The other reason is going to have a much more common flavor to you of epigenetics. And to explain that, I'm going to draw another chromosome territory here to give myself a little bit of room to talk about something here. I'm still going to draw this loop out here, the rest of the DNA in here, and just up a little of these nuclear organizers, these tandem repeats, we see large areas of heterochromatin. So large that it almost forms, not almost, when you look at it, you kind of see this ring of heterochromatin all around here. Remember, this is three-dimensional, so there's going to be some nuclear territories on top here. And at the base here are all these heterochromatin regions. It's so effective that dyes that stain DNA can't even get inside of the nucleolus to stain these loops of DNA. We know that here, in these regions of heterochromatin, that our good friend from before, H3K9, is methylated. Remember, H3K9 is the site when it's methylated, it turns off gene activity and forms heterochromatin. And then we have H3K27, which is one we haven't talked about a lot, and it is also methylated. Together, this methylation pattern in these regions right adjacent to the nuclear organizers allows heterochromatin to form. So here I'm going to put, for the second point, I'm just going to write heterochromatin. To demonstrate its importance, I want to say one thing here. We've talked before about this complex called SIR2. To remind you, it's a histone deacetylase. When this is mutated, we see these ribosomal DNA, where the genes are at for ribosomal RNAs, are unstable. They're not transcribed as you would predict. And the nucleolus is disorganized to the point where it doesn't form this nicely and easily identified structure here with these loops inside of it. So forming these epigenetic marks here to form or establish the heterochromatin is very important in maintaining and forming this nucleolus. Now what I want to do is talk about the rDNA, the genes in here that make ribosomal RNAs. Okay, so let's talk about those genes and how they're going to be turned on or off. One clear way that they'll be kept turned off is if the DNA is methylated, just like we've talked about before in these CPG repeats. So this would be off. But that's not all that requires to keep it off. If you want it to maintain its permanent off state here, or a more firmly established off state, you need this protein called, or this complex called NORC. Associated with this complex we have DNA methyltransferases for the DNA being methylated. We have histone deacetylases, and we have histone methyltransferases. Together, this complex will come along here. It will keep the DNA methylated at that CPG repeat area, but it will also, together, these DNA methyltransferases, the histone deacetylases, and the methyltransferases will turn this gene here, or keep this gene, off. Now, to make it active, what has to happen is this protein complex called CSB comes along, and CSB will do a few things. It will methylate H3K4, and it'll add 3 methyl groups to it, which remember is the active state. And it will acetylate H3K9, so that will come along here to cause the gene within this region to be activated or make it on. So the NORC will epigenetically mark this region to keep it off. The CSB will come along here and turn it on. Having these large complexes come in here helps establish that epigenetic mark that is strong enough to be inheritable. Okay, that is all I'm going to say for now on this chapter. But if you have any questions at all, please make sure you come see me. Bye.